Greetings of the day. My name is Raza Muhammad, and I'm a PhD candidate at Chulalongkorn University in Bangkok, Thailand. Today, I would like to talk about the gut microbiome of long-tailed macaques, a nexus of environment, diet, and host genetics. Macaca fascicularis, or long-tailed macaque, is one of the non-human primates that is widely distributed throughout Southeast Asia and is currently being used in biomedical research. Based on morphological characteristics and geographical distribution, this species has been divided into 10 subspecies. Among these 10 subspecies, Macaca fascicularis aurea or MFA is strikingly important because of their stone tool use behavior during foraging to access in-case food items such as oysters or nuts. MFA originates in Myanmar and is distributed southeastwardly in Thailand through the Margui archipelago. MFA lives in close contact with another subspecies of long-tailed macaque that is Macaca fascicularis or MFF. However, MFF has never been reported to use stone tool in nature nor in captivity upon training. Thus, previous studies indicated that distinctive genetics characteristics between the two subspecies. Thus, it has been hypothesized that different genetics might be one of the factors that led to the emergence of stone tool use in MFA, but not in the other subspecies. Moreover, MFA primarily inhabited coastal and estuary habitats to a greater extent than the overall MFF populations. With respect to the stone tool use behavior being found only in MFA, it has also been proposed that it might be due to the differences in their habitat types, which in turn could reflect the different types of food availability in their natural habitats. Therefore, we hypothesize that variations in the microbiomes between the two subspecies of MFA and MFF might correspond to their habitat type and dietary preferences in association with the performance of their stone tool use behavior. This lead to the research question that do differences in bacterial microbiomes between MFA and MFF relate to habitat, dietary preferences, and their stone tool use behavior. Two populations, each of MFF and MFA residing on the island and in mangrove forest, were selected, as you can see in the photo. And the food types that were consumed by the monkeys were identified either by direct observations or by leftovers of food items, which the monkeys finished eating. Fecal samples were non-invasively collected in the field by fecal swab method, as you can see in the photo, and the DNA was extracted and the full-length 16S rRNA gene was amplified and finally sequenced on the MinION platform. The results showed that the two populations of MFF were receiving anthropogenic food from tourists and local people, while the MFA populations were solely relying on their natural habitats. Based on the diversity analysis using Chow-1 and Shannon indices, it showed that the MFF populations who were receiving anthropogenic foods in addition to their natural food exhibited higher alpha diversity as compared to the MFF populations. While the beta diversity showed that the MFA mangrove population exhibits distinct bacteriomes. Now here a question arises that why do the alpha diversity is higher in MFF populations than MFA population? And it can be seen in a previous paper that has also reported that the provisioning leads to the higher alpha diversity. And it might be due to the introduction of the new foods that apart from the natural foods in their natural habitats, these provision macaques could also access additional food regularly provided by humans. And it could possibly lead to the higher diversity of their microbiome. Based on taxonomic classification, the top taxa at the phylum level were Firmicutes and Bacteroidetes, while the genus, the macaques, were highly enriched in Prevotella, which lead to the carbohydrates and the fibers. While at the species level, these populations show the higher diversity of Clostridium difficile, which can lead to the infection of Clostridium difficile by the Clostridium sensitis taxa. Moreover, the left-C analysis revealed that the MFF island population exhibits higher bacterial taxas as compared to the other populations of the MFF and the MFA at mangrove and island populations. To conclude, these results suggest that the diet, which includes the natural food, anthropogenic food, and the foraging behavior, which is the stone tool use behavior, and also the host genetics, because the two subspecies have distinct genetics. And the third one is the environment, which lead to the differences in the habitat types, have an influence on the gut microbiota of these long-tailed macaques. And as I mentioned, that the two populations were receiving foods from the humans. One of them were receiving from the tourists because some of the populations are living on the islands, which is an tourist attraction for the tourists, which regularly feeding by the humans, and also the local population also feeding the monkeys, which leads to the macaque dependence on their provision food. Thus, it has been advised and suggested that the monkeys should not be feeded in their natural habitat because it could affect their natural behavior. And finally, I would like to acknowledge all the people who supported and contributed in this work, and thank you for your kind attention.